name is Paul Ebner. I'm an associate professor in animal sciences in Purdue University. I grew up in Livonia, Michigan, which is not an ag city. It's a suburb of Detroit. And I got into agriculture when I joined the Peace Corps after I graduated from college. I work in microbiology. We try and come up with different technologies to get rid of pathogens like salmonella and E. coli, either in the animal, like before it goes to a processing facility. What we're currently working on is coming up with different solutions to challenges in food safety. So the CDC, the Centers for Disease Control, come out with these huge numbers of foodborne illnesses each year. A lot of them can be associated with meat products or animal products, so we try and come up with different ways that we can get rid of those pathogens.
My name is Luis. I'm from Brazil. I'm working with Microphages on Purdue Animal Science Department. In Brazil, my major degree is on biomedical science, and I like the microbiology class, and I'm doing my master's. I did my master's on food microbiology. So we are testing the ground pork bacteriophages against salmonella.
We concentrate on a couple stages. One's called the pre-harvest stage, so that's on the farm. So coming up with things that you can either feed to an animal or put in the animal's feed. And we also look at the post-harvest, and that's actually during processing. Well, one of the technologies that we work with is called phage therapy. Phages or bacteriophages are actually viruses of bacteria. So just like you have a virus that gives you a cold or flu, bacteria have viruses that infect them. So bacteriophages, how they work is they're very specific for specific bacteria. So say if it's a salmonella bacteriophage, it finds salmonella, attaches, and if you see pictures of them, they look like little rocket or spaceships kind of attaching to a bacterial cell, and they inject their own DNA, use the whole bacteria to make more phages, and in doing so, they cause that bacterial cell to lyse or explode. So what we try and do is we recognize that there's an antibacterial property of bacteriophages, so we try and harness that. And we've been pretty successful with that. We've developed even a way that we can microencapsulate them, which is a fancy way for saying it's wrapped in these little fat balls. And we can put it, with that we put it in swine feed, we directly fed it, and it works. It cuts down on the transmission of salmonella from pig to pig. So the overall goal eventually would be to attempt to commercialize this. And so we can get it into the hands of producers or food processors, because it's a technology that we know it works. These are natural products, and we're just taking what's happening in nature and kind of concentrating it so we can get rid of these specific bacteria.